Hello and welcome to a 20-minute talk about the membrane. I'm going to have the membrane talk with you. Now, as we well remember from the beginning part of this class, that the plasma membrane basically defines what's living. Everything that's on the inside of that membrane is living. Everything on the outside is basically non-living. And it's the membrane that selects what goes in and out. This graphic here I like a lot because it's showing you the phospholipid, well, actually this is the hydrophilic end and this is the hydrophobic end of the lipids. This is showing you a crush of water on the outside, a crush of water on the inside, very few spaces in there. This is showing you some type of aquaporin, so we have a protein that goes through here, and we see these kind of water molecules kind of running through. And so while the membrane, for the most part, like in these areas, is very good at preventing water from coming through, membranes in these areas can leak water through pretty much. So we'll talk about that. Again, here's another view from straight up forward. Again, these would be the hydrophobic regions that would anchor this particular protein into a membrane, and then we have this hole in the middle here, and this particular one is a potassium channel, so that's just big enough for potassium to move through, but water should be able to move through it as well. I don't have my video up and working. Again, here's the movement of water through an aquaporin. This is not showing the membranes on these parts here, but basically this water just kind of pours through this area to go into the cell. We do want to know about the membranes. We do need to know that they are made up of phospholipids, which are amphipathic molecules. Know what those are. The fluid mosaic model is the latest, greatest, and best way that we have of describing how our membrane works, so you should know it. These membranes are about 5 to 8 nanometers thick, and so think about a cell like E. coli being basically 1 micrometer across. So not a lot of that structure is taken up by the plasma membrane. Again, that was a little bit of the history of kind of our thought on what membranes were. I covered it a little bit in class, and I'll do it. So basically we have here the structure. These are what we think of the phospholipids. So we have a hydrophilic head and a hydrophobic fatty acid. These are made from the fatty acid tails, and if we look at them, they look something a lot like this. And again, you should be able to tell me where the charged are and whether they are saturated, or in this case here, this guy here is unsaturated. Again, when we look at it in a fluid mosaic model, and we had fun with a couple of students walking around in class, we have these phospholipid molecules that form this kind of bilayer here, and integrated into that are some proteins. And so these proteins here are going to have some hydrophobic regions on the outside of this protein, so it can hang out in here, and we'll have some hydrophilic regions over here, so it can associate it with something else. And so this phospholipid bilayer is not just kind of a soap bubble that we thought before, and things get really complicated, okay? And so we'll cover this several times. But again, even though it's relatively small, there's lots of support. And so these membranes are connected to cytoskeletons, the extracellular membrane, so that they are pretty stiff and pretty rigid, and they're not going to fall apart easily. And this one here I want to talk about is basically what gave us evidence for this structure that we have. We had this experiment called freeze fracturing, where we froze a cell and we tried to break it right at the plasma membrane. Incredibly small, incredibly detailed, incredibly good work. And so again, we can take a look at the different proteins and the formats of the different membranes that we have. Again, the phospholipid bilayer, these cells can switch with one another. The phospholipid can rotate. They flip-flop relatively few times, so maybe once a month. Unsaturated hydrocarbons form kinks and tails and makes it more fluid at lower temperatures. Saturated hydrocarbon tails makes it more viscous at higher temperatures. And then cholesterol does dual duties in that the cholesterol molecule itself can prevent the movement of this, but it also introduced kind of like this random structure so that it allows more fluidity at kind of lower membranes. Again, another experiment that showed us this is basically we took a mouse cell and a human cell and labeled their different mouse proteins and human proteins, and when we fused those cells together, those proteins within an hour moved all over the place. And so this really shows us that this membrane is not static. It's not just a piece of marble that's up there. It's working, and it's this fluid state that allows the membrane to work. So when things get too cold, your membranes start working. I guess you got to worry about other things when it gets too cold, but again, be aware of kind of what happens with the structure of a membrane at different temperatures and why we have saturated and unsaturated. And again, here this is looking at that extracellular matrix. We talked a little bit about this in class. For this matrix to be made, these parts are exported from the cell and released out here, and enzymatic reactions occur here. We have lots of proteins. We talked about the globular structure of proteins. These proteins, we could also have what we call carbohydrates attached to the protein. So we have glycoproteins. Many proteins are modified in the Golgi apparatus to become glycoproteins. And then there are also glycolipids that we see as well. And then again, a student in class was possibly talking about deoxyribonucleotides as well, and so lipid nucleotides. I haven't really heard about those. Again, the two types of proteins that you want to know are the peripheral proteins that are mainly attached to the outside, the integral proteins that penetrate the hydrophobic core. They may go through all the way through the side, so again, from one side to another. In this case here, we have both our N-terminus and our actually no, amino terminus is kind of on this side. And so we'll talk about that a little bit later. You do want to know the major functions of, heck, I'd almost just say enzymes, but it's also the major function of the proteins. Main one is transport, moving stuff from one side to another, enzymatic activity, signal transduction, cell-cell recognition, intercellular joining, and attachment to the cytoskeleton. And I made a protein, and Chris Case made fun of my protein being a sticky integrin. Anyways, in transport, we have these channels that will facilitate transport, and enzymes, again, when you're doing a pathway, we want to go from red triangle to green circle. Again, if we put enzymes right next to one another, that's going to facilitate that movement. Signal transduction, we can have a signal on the outside of the cell that can move on to the inside of the cell and cause some reaction to occur. And cell-to-cell recognition, which we've seen in our immune system, you will have cells that kind of recognize certain glycoproteins and kind of act like barcodes, or they act like secret handshakes, so that the cells will recognize one another. Intercellular joining, and again, when we talk about our gap junctions or our tight junctions, allow two cells to kind of interact with one another. Because remember, these are the same charge, and they're basically going to repel one another. So how do we stick cells together? How do we make cells sticky to one another? And again, lastly, it's looking at that extracellular matrix. And so we covered these actually a little bit. Again, how does the membrane carbohydrate play in cell-cell recognition? It's a cell recognize each other by binding surface molecules, often carbohydrates, on the plasma membrane. Again, glycolipids and glycoproteins. And this is made up in the Golgi apparatus, so we can have some symmetry in here. And so in the case here, we have a particular molecule that we want to be expressed on the outer surface protein. It has to be on the inner surface, and we see as this kind of conveyor belt goes through and the endomembrane complex, we see that these proteins are carried through too. And so a lot of times those proteins are modified in order to do what we need to do. Selective permeability is basically talking about the transport proteins. We do need to know what diffusion is, and so here's a graphic of diffusion showing lots of molecules in a solvent on one side, solute in a solvent, and then what happens is over time we reach equilibrium, where the exit going out the same as the entrance going in. Again, if we have two different sets of molecules, we can have them going in the opposite direction, and that will still work. Again, the energy that's driving this is basically Brownian motion from basically the temperature that basically everything we know that's alive has. Substances move down their concentration gradients, the difference in concentration of substance from one area to another. So the higher the concentration, the less likely that material is going to move towards that situation. Osmosis is the opposite. It's diffusion of water. So if we have a selectively permeable membrane and we put some sugar inside of there, what's going to happen is the water is going to move to kind of help hydrate the sugar and make up, and that we can actually use this to do work. Tonicity is the ability of a water solution of water to cause a cell to gain and lose water. Isotonic is where the solute concentration is the same as the inside of the cell. Hypertonic, solute concentration of water is greater than that inside the cell. And then hypotonic, where the cell concentration is less than the inside the cell. Solute concentration in the cell will gain this particular water. And so, again, if we were to take water and put it in a hypotonic solution, in this case it's just pure water, it would fill up and lyse. Plant cells tend to not do so, but we'll talk about it a little bit later. In an isotonic solution, the plasma membrane will actually remain the same and pretty healthy and normal. In a hypertonic solution, we actually see an exit of water through this, and you'll see the cytoskeleton structures in there. And an osmoregulation is basically the control of water, and there's a number of different ways that organisms have controlling osmoregulation. And in tomorrow's lab, you're going to be actually looking at a particular organism that has a contractile vacuole, which you'll see it kind of pushing the water out. It's kind of like a boat. If your pumps work faster and pump water out faster than it comes in, then you're going to stay afloat. Turgid and limp, you need to know these are referring to plants. Plasmolysis is also referring to plants, and facilitate diffusion. And so basically what's going on facilitate diffusion is where you have a membrane channel that's controlling which molecules kind of come through, a lot like a doorway. Again, you don't necessarily need to use this doorway. You can use other doorways that possibly fit as well, and we'll get into that a little bit. Protein. And so how it works in active transport, an example here is a sodium-potassium pump, is that we have basically under the right conditions, okay, the sodium is going to bind to the inside of the pump, and we will hydrolyze the protein and release the materials, and then we get it all over again. I want to show you that little animation. So why would we want to do something like that? Well, basically this energy difference allows us to use it, attach it to do other work. So when we can't use ATP directly to do something, we can set up one of these pumps in order to do it. The sodium-potassium pump is not the only pump. We use a proton pump, and this is an example of this here, and so this can generate a really good co-transport is where basically you need to use up some of those ions in order to pump things like sucrose into the cell. Exocytosis, when transport vesicles migrate to the membrane, fuse with it and release, and then endocytosis is the opposite. And so here we need to look at this, and so here we have phagocytosis, which is a relatively large structure. Pinocytosis is relatively small. Receptor-mediated endocytosis we tend to see with our antibodies. In this case here, it's just membrane. You know, we're trying to look for the different ones, membrane pit-coated, so that may be more something kind of unique. Okay. And I just want to say it's the end of the talk. Thank you very much. And